introduce vectors and matrices. Last week, we also talked about 10x genomics, how the barcoding system is working. So you can get information from individual cells by utilizing this barcoding system. Today we will, after introduce those important concepts in lineage algebra, it's a very quick 101, the foundation, okay? Then we will move to 10x genomic cell ranger and see how we can read the report from the cell ranger. So because cell ranger will help you to read information out of your FASTQ files. That's after alignment to a reference genome. We did this in the cloud environment. Then you will get gene expression matrix and also with the QC report. So we are going to spend some time to read this QC report, see whether by looking at this report, we will have an idea whether your experiment for that particular sample is successful. How many genes, how many cells has been measured for its expression, whether this saturation curve makes sense. All right. So let's see if I ask you, so what is a matrix? What's on your mind first? So anyone say, okay, I have a matrix. What do you consider, what jump into your mind first? So matrix is, you may say, okay, it's a spreadsheet in the Excel. So it's a table, have rows and columns. What else? It's a table. Matrix can also, if it's a squared matrix, it can describe the similarity between two objects. So this object could be a cell, could be anything with described with a string variable. It could be a relationship between variables. So graph, the network, can also be represented as a matrix. So matrix could be a graph, could be a description, pairwise the relationship between objects. So I will use data points a lot. It's a collection of information. It's a table. It's a name of a movie, a matrix. It's a movie. So today, after this lecture, after the first half of the lecture, I will give you an impression that the matrix, especially the squared matrix, can be used as an operator. So this operator can do something to the data points to either rotate it or change the magnitude of the representation of this given data points. So right now you may have no idea why the matrix could be an operator. So operator is just like a plus. Okay, you do multiplication, so put two variables, two numbers. Matrix itself can be an operator. This operator can do rotation, can change the magnitude of the vector length. how to say, stretch. I don't know if this is a correct word. Stretch, make some longer. Is that the right spelling? Make some longer or shorter or compression. So anyway, if you have no idea about what it is, so after the lecture, I hope you can consider matrix, especially the squared matrix. As an operator. So that's the thing we want to learn. And then once you have this concept on your mind, you will get some idea about what's the eigenvalue, eigenvector. It's very important. I'm glad that you didn't miss this class because, as I said last lecture, linear algebra is so important, essential for machine learning. So machine learning, it's start with all the chip revolution, okay? So I have a slide. So you see this last week. And you see this Moore's law last week. This is an AI revolution, but I didn't show you this picture. So this basically tells you all the machine we are using, like this one, as all the high-performance computer support this language model or this AI revolution. It's based on transistors, okay? Transistors to make this machine, adding machines. The only operation that machine can do is add up things, okay? Using the binary representation. So you encode A as 0, 1, 0, 0, 0, 1. So by adding a value, so you make a operation. So all the machine can do, all the foundation, hardware foundation of this information technology is based on adding machine. So everything is just add up, nothing else. So multiplication is just you add multiple times, that's multiplication. You can think any operations you can simulate, you can generate, you can create, you can store in the computer. Now it's adding machine, okay? There's no difference between your current machine operation compared to the abacus, which 500 BC, we have recorded the earliest one. So that's the information I want to give to you. So this is a part of this student group talk I gave at a different time. So come back to this. So so how adding machine can do this? Can optimize the distance between all the points to this straight line. So this is optimization. We try to find a line which minimize the difference for all the points to the line. So this is part of the PCA. So I show you this teapot. We are going to use this as an example to show you. So PCA is try to find an angle to visualize your object. So this object is 3D. But if I see, okay, you only take one picture for this 3D object, then you have to very carefully select the angle which you will take this picture from this particular angle. You don't want to take a picture from this end, because in that case you will see a very different object which cannot give you the full the best feature of your object. So you select this particular angle because you want to rotate this object to achieve, to represent, to show people the most features out of it. Okay, this feature is given to by the separation of the data points. If you consider all data points on this surface, you want to show the maximized variability representation. So the point from here to here basically maximize the difference between points of the data on this surface of this teapot. So you will see, okay, how can we achieve something like this by using adding machine? We only do addition in this computer. How can we search, rotate, and look at, search again until we found this right angle to visualize this object? The value of this talk, I believe, is through this face-to-face, intensive 2 hours interaction between you and me. Otherwise, there are so many books, so many online materials you can get for linear algebra. You may just not have a patience or time to read through, to look at those YouTube. But this intensive interaction between you and me in these 2 hours gave you very quick idea to get into the core of linear algebra, which is the core for machine learning, for everything you will learn from now. As I said, after lecture, you get the squared matrix is operator. That's all we want to achieve. I know the biology student, what's on your mind, so I understand. So it's very important to come here to take a lecture. You can have a video, YouTube. There are many, many different resources, as I said. Especially in this generative AI age, you can have customized tutor for yourself, of course. Yeah, but still, this may be the last couple of years that we do this face-to-face. Maybe the education system will change, okay? I appreciate your time now. So vector, it's, so we separate variables, like in MATLAB. We call scalar, so this is just single value numbers. So you can have 5, so this is a variable, which is a scalar. The vector is a collection of scalars, so you can have 3, 4, 1.2, so something like that. Okay, you can make it longer. So scalar, this can be 1 times 1 vector. If I say, okay, my vector is 1 by 1, or my matrix 1 by 1, that's a scalar. So that's also the concept in MATLAB and other languages as well. So vector is 1 by n, or n by 1. So this vertical is n by 1. So that's a vertical. Horizontal, okay, you have a n by, so this would be 3, 4, 1.2. You can do this. In data science, in machine learning, we consider vertical vector by default. If someone in the textbook for the linear algebra in the data science, they put a small a as a vector, and it does not tell you whether A is vertical or horizontal, so by default it's considered as a vertical. So this is also a vertical, is also a data point. Because this vector A holds the information of an abstract object, which will represented by the elements or numbers in this vector. So those vectors describe the object A, describe this data point, and those numbers are the variables. So variables, a collection of variables with multi-dimension. So this case, if I only have 3 numbers, the 3D representation of A as a data point. And this A, if you do the transposition, from the vertical become horizontal. Okay, this is an operation. Sometimes they put AT, so there's a transpose. It's a transpose operation to put a vertical vector into horizontal, or you can do that again. You transit back, okay? Transform, transpose. All right, so now you have idea. This is very simple. Don't have to remember, right? I know in intermediate school, people study this. My daughter in intermediate school, they know this. Matrix. Matrix, I will first consider it as a collection of vectors or data points. Okay, so if A is a gene, is a cell described by 3 genes expression, then I can collect a lot of cells and put them into a vector, okay? Into a matrix. So this M, we can have first column, second column. So this will be the first data point. And then you concatenate the next one. So this will be N by M, so whatever. So this N greater than 1, M greater than 1. If there's 1, it collapses into a vector, okay? So on here is a row vector or column vector. So it is horizontal, vertical, same thing. Matrix there. As I said, if you have 3 numbers, 3 variables, so this will be variable 1. Variable 2, 3. So 3 variables to define a data point. So this gave you the introduction of the 3D position of this particular object. So the 3 here, 4 here, 1 point is somewhere here, okay? So by its coordinates, you can put this data point in the 3D space because you have 3 variables, so there's a 3D. To draw the vector, you put a point, start from origin, so this will be 0, 0, 0. In the 3D, you point to this data point, which defined by the 3 variable values. This will be x, y, z. Now you need to use your imagination. If this goes to n, goes to n, then you have variable number n. So you have to imagine n dimension higher than 3D, so n dimension space. You have to imagine, okay, this A is located in a ND space in that position, but you still start from an origin and draw a line to there, point to it, to represent this vector. Okay? Start from 1, 1, 1. If you have a n dimension, you have 0, 0, 0, 0, 0, n numbers of 0 as the origin. So that is the geometry of the data points. So always think about you have vector point to a place. Okay? This vector is either direction is number 1, the most important. So point to which direction. Number 2 is how long this vector is. Okay, so some vector is very short because the number is small. Some vector will long, okay? And so we compute the high dimensional Euclidean distance or whatever. So you compute the distance from the data points to the origin. So this as a magnitude. So D, so how long this vector is. So it's, we'll have a name, okay, later. Now we start to learn some operations. I don't want to spend too long here. It's so easy, right? Your scalar, you can multiplicate this by a number. If you multiply by 2, this becomes 6, 8, 2.4. So you double the length of your vector by multiplicate or scale it longer or shorter. You can do addition. So if you have 2 vectors, if you have 2 vectors, so this is A, then you have a vector B. So A is represented by 3 numbers here. B is also represented by 3 numbers here. So A plus B, you can just add them up. Element which together. So what that means is you add the portion of the A. So this number defines how long A projected on the x-axis. This first number in B represents how long the B projected on the x-axis. So you add them up, then you make a longer vector to that direction. And also you do the same thing for the y, z at the end, you get a longer version of this. So this new vector, a plus b, will still in this 3D space because you only add up the same number of the variables correspondingly. So your summation of these two vectors will be still in this 3D space. But the longer depends on how long their summation is. So the geometric representation will be in this place. Okay, you add them up in 3D. Same thing happened in ND. So if you have a cell which described by 6,000 of genes, so you will have a cell number A, cell A, which have a lot of genes to describe this particular cell. Then you can have a cell B, which also have 6,000 genes, 6,000 numbers. You can add them up. You create a new cell, which is A plus B. That cell will be something like this, okay? Cell A is here, cell B is here. I have never think about it to do addition of two cells to make what that means in the in the real world. But you can do this operation very easily. This could be a duplex, okay, two cells captured together. One is a T cell, another macrophage, and this droplet didn't separate them very clearly, but capture them together and measure the gene expression of both. Maybe in that case, you get A and B, and you get a duplex cell. So sometimes people describe a vector which not start from an origin, but you can move, for example, this vector from P to Q. So in that case, this represents the same vector like this if you consider P as an origin. So what you will do is move this coordinate system to that direction. Depends on the position of the P, you move your origin to that P and go up and make the P is origin. So that vector represents the same thing. So you just do this operation. You get the PQ position, PQ vector. So that's what I described, the addition operation between two vectors. So this is the subtraction of two vectors. So if you do U minus V, you will get this vector. Okay? This vector represents U minus V, or V minus U, which is this one as well. As I said, you can move this to the origin, which you get this. So when you do this two vector A minus B, you get the distance between two vectors, two data points. The length of this is the distance of the two data points. No matter how many dimensions you are dealing with, so n dimension or n plus 1, when you have two vectors, these two vectors project into the space. One's here and the other here. all you have to do if you want to calculate the distance between these two data points, you just do this subtraction operation. You use vector A minus vector B. You get a vector which in between of them. So the magnitude of this is the distance between two data points. That's why we really like to represent information into a vector, into a matrix. Because matrix is a collection of vectors. This vector 1, vector 2, vector 3. You can have m different vectors. Now what I can do is to use vector 1 minus vector 2. So this will be compare two data points and calculate their distance. So this will be, see, let's name this A, B, C, D. Different name of these data points. So the first one will be A minus B. So this will help you to calculate the difference between these 2 points. You can A minus C, A minus D, and then you B minus C. You can do this pairwise difference between vectors. Is that easy to calculate difference between vectors? Not too hard. You just match them and do the difference between, do the subtraction between all the elements in there. So we can very quickly, if you do this pairwise difference vector subtraction, you can very quickly get the distance between data points you collected. Here you collect m different data points. You just use this pairwise subtraction. You can very quickly get a distance matrix. You compute their pairwise difference very easily. Okay. Let's talk about production, the multiplication of two vectors, okay? I think I will jump. This is so important because I want to do matrix to matrix multiplication first. Then we come back to vector times vector, okay? Because vector multiplied by another vector is a simplified version of the matrix multiplication. Because vector, if you reduce the dimension, one of the dimension to 1, this becomes vector. So if you know how to do matrix to matrix multiplication, you will automatically know how to do the vector to vector multiplication, right? And the meaning of this is to help you to find, if you do this to vector multiplication, you will be able to find the angle between them. So if one vector point to here, another point to here, the angle is greater than 90 degrees, you know there goes to different direction. If this angle is small, although the magnitude is matters, but if you normalize it, come back to the same magnitude. So the direction becomes very important. So the vector multiplied, the product, called inner product, will help you to get the angle between two vectors, not a distance, okay? So here, subtraction help you to get the distance between them. Well, here, the production, the inner production, the dot product of two vectors give you the directionality, whether these two data points point to the same direction in this high dimensional space. Depends on how many variables you use to describe the vector or data. So we are going to jump and come back to this page later. We are going to jump to matrix multiplication. Is that okay? Any questions very quickly? So we will come back to this page. If you have time, if you want to read, you can go ahead, but we will come back to this concept, the dot product and also the angle between two vectors. Now, we will study the magnitude of the length of vector. So this is a name called a norm of vector. So this vector, this norm, so we will still use, so everything you already know. So to compute how long this vector is, so you just need to calculate the distance between this A point. So my example is 1 for 1.2. You just use Pythagoras theorem, okay? The distance between this A point to 0, 0, 0. So this is description of the variable x1, x2, see, x, y, z. So x, y, z, three values for their coordinates in the 3D. So all you have to do is plus y squared, z squared, then square root. So this is the distance, right? This is the distance from origin to A. So we gave this name. So this distance is a norm of A as a vector. So the norm is a scalar, it's a single value. Sometimes we write in this way with a bar. So A is a vector, contains many numbers, small numbers, okay, individual numbers to describe the A position in the space. But if you put A, then put a two vertical bar around it, just like this. That's a scalar, it's a number. A single number indicates how long this vector is from the origin to the end of this vector, okay? So it's a Pythagoras theorem, so you get squared. So that's why sometimes it's also called L2 norm. L2 norm indicate this is a distance using this Pythagoras theorem to calculate. There are also L1 norm, for example, of a vector. L1 norm is just everything added up. So L1 norm for this will be 3 plus 4 plus 1.2. Another concept is the unit circle, okay? Let's use this as a 2D as an example. So on this circle, all the data points have the same norm. So the vector, the norm of all the vector in here is 1. So this unit circle will define all the possible direction you can point to in this 2D space with a unit vector which the length always 1. So if you have a longer version of this, so for example, you have A. What you can do is to use A divided by itself, the magnitude. You will get a uniform version, how I say it. So you basically you normalize the length. You push it back onto this unit circle. Okay. We use unit circle to give you the points, the directionality. Don't give you information about the magnitude, only care about where to go. So the position here, for example, is 1, 0. Here is 0, 1. Okay. Here is 0, -1. You see these 2 points, these 2 vectors on the unit circle will define this coordinate system. Okay, if I gave you a set of 1, 0, 0, 1. So this is this vector, and this is this vector. Remember, it's vertical. So this can give you a definition. These 2 vectors give you a coordinate system definition, which is your x-axis goes this way, your y-axis goes that way. You can use this and that, which are slightly different number of 2 vectors to represent another set of coordinate system, as long as these 2 vectors are orthogonal to each other. We'll come back to this later. So now I need to spend the time to show you how to do matrix operation. We don't have to do transposition because it's very easy, so you just rotate your rows into columns become rows. In MATLAB, it's just you have a x, then you just put a apostrophe operation in there. This will be, the matrix will be transposed. Addition operation requires 2 matrix have a same dimension. So if you have a matrix 4 by 5, another matrix also have to be 4 by 5, then you can do addition. you just do every element inside corresponding position, just add them up. Scalar operation, multiplication, easy, okay? So this is transposition. Transpose, okay? We don't have to explain. Identity matrix means only this diagonal position are the 1 and all the others are zeros. So identity matrix has to be a squared matrix. Same numbers of rows and columns. Addition operation, adding. So you have a matrix A and a B, the same as B plus A, or you do this. You can group A and B first, and then plus C, or do B and C first. So those are the operation sequences, doesn't matter in the matrix addition. Scalar multiplication, so you times 2 and to a matrix, then every element in there get multiplication scaled by 2. Easy. Okay, so now we are going to do matrix multiplication. So first, if you have no idea, you can do, if you have 2 matrices in the same dimension, you can do element by element, just like you add one matrix to another. But when we talk about matrix multiplication in general, we are not doing that way. Not element by element. So this operation is so important. It's underlined every, almost every operations in the computer science process. Okay. So the best example I can find is like this. This is you. First, we define a matrix A. There's a meaning for this matrix. This is you and the row, and that's your roommate. And you roommate want to make 3 different kinds of sandwich. So the first column, so the A is a matrix of 2 by 3, okay? So A is a matrix. B. So the meaning is that you want to make 4 sandwich type 1 sandwich, and the type 2 sandwich you want to make 5 of it. You want to make 3 sandwich of type 3. So you have 3 different types. Then your roommate want to make a 3 type 1, 3 type 2, 6 type 3. And the B, so A has dimension, so this A will be 2 by 3. B has dimension 3 by 6. So the B is 3 rows, okay, 2, 2, 2, 4, 4. In this example, B is also has a meaning. It means the B is B is what? The butter. B is bread, so you need the bread, and the P is the unit of peanut butter. So J is a jam, jelly, something, jelly, hams, tomato, ham, and also cheese and tomato. So each column is kind of a gradients you want to make sandwich. So for the type 1 sandwich, you need bread. For the type 2 sandwich by rows, you need bread. For sandwich C, the third type, you need bread. Bread is essential, so every sandwich has a bread in there. But you can pick either you want peanut butter, jelly, or not. Depends on which type of sandwich you want to make. Then you want to put tomato, you want to put cheese on it. So that's the gradients to make them. So these two matrices can be multiplied. So A times B get C, okay? So this multiplication can be done. The reason is the 3 and the 3, because they have shared this dimensionality. So the dimension for this is 2 by 3, and here the 3 by 6. These two numbers, I call it inner dimension to match. So if you have two matrices, when they multiply, you have to make sure their inner dimension match together. Should be the same dimension. If not match, for example, here you have 4 different kinds of sandwich instead of 3. And here you only give a component, the recipe for 3 sandwiches. You cannot do this multiplication. You have to match the type, number of types of sandwiches. So how can we do this multiplication to get a C? So normally I will do is to put three. So this will at the end give you the C, which have a dimension 2 by 6. Okay. I will see. I will put. So So how A is 2 by 3. So this is a 2 by 3. I will put 3 by 6. So that will be 3 by 6. So I can arrange these two matrices, one put here, another put there. The reason I want to do this, I want to let them to match. So there are 3 numbers here, and match with the 3 numbers here. Oh, no. Okay, so I can do operation between these. You see, this is a rectangular, and this is a rectangular. I will get another rectangular, which is 2 by 6. That's the way I just put, arrange the location, the outline, the position of these two matrices, and so I can guarantee I will get the correct results at the end. What you will do is to look at the first 3 numbers. Our first 3 numbers here is 2, 2, 2. Remember, they are bred. Okay? This is a B. So if I want to make type 1 sandwich, I need 2 bread, sandwich 2. So now I just need to multiplicate how many sandwich each. You multiply it by the number here, and the summation, then you get the number. Add them together. The times first, each element times each element here, and then summation. You get 24 in this position. You get another 24 on the second row. What that means is you need to buy 24 bread, because you want to make 4, 5, 3 different kinds. When you want to make a 4, you need 8 here. When you for the 5, you need 10 here, 10 bread. You need 6 here. So sum up, you have to go out to buy 24 bread. And your roommate, depends on his or her numbers of sandwiches you want to make. These numbers, you multiply these 3 and sum together, also need 24 bread. So all you have to do is just get the values, 3 values here for each row, multiply it individually to the 3 numbers in each row here, and then sum it, put them together, you calculate. At the end, you get this multiplication, the results, to indicate, for example, you need to buy 15 slices of ham. Well, your roommate only need to buy 9 in order to make 3 sandwiches. Use that number, okay? The reason you need to buy more ham because you have more type 2 sandwiches. Type 2 sandwiches require ham. Okay. Okay, now you know how to do matrix multiplication. Number 1, you need to remember is match of the inner dimension. between two matrices. What that means is you have a matrix 2 by 3. You can multiply it with another matrix 3 by something else. So 3, 3 has to be the same. 3 equals same. So that's a number 1. Number 2, you need to put them into this position. Okay? Make sure you can get the square, the rectangular, in the right dimension at the end. Okay? Put them in right position. Three, just do this matched multiplication, element by element, then summation. Okay? Element based then so you do this element-based multiplication, then summation, and put that position here, corresponding position, until you fill up all the position in your A times B. Okay. Now you know what that means. So multiplication of matrix means you mix some different components in certain weight, or so you mix these components or gradients in some way based on your how to say, loading, or the proportion you want to make. So it's a kind of a mix components together by certain ratio, by certain amount, to get the total C. So you go out to buy those components as a bulk, okay, together. Now I have a question for you. If you know the matrix C, you have a 2 and a 6. Can you reconstruct A and B? So the reversed process. If I give you, say I have a matrix with 24 24, 16, 18. If I have this, I give it to you. I say, okay, we go out to buy 24 bread. You guys buy 24 bread. We buy 16 peanut butter. I gave you the shopping list of your 2. Is that possible for you to figure out what your roommates want to make, yourself want to make? So if you have a C, can you reconstruct A and B? Now I want you to think. This is a gene expression program. So for example, this will be a cell which have 3 genes to help translation, transcription, and apoptosis, and something else. So you have a different gene expression program encoded by those genes expression. Then you have 2 different type of cells. And the cell 1 will have some of this function, 4 part, some of this function, some of this function, so mixed function. They all together, but you see, okay, one gene may be involved in different program, but at the end, you only measure this cell expression of that gene, which is C. But you want to figure out how many different gene expression program in that cell in what proportion. So the opposite way to do this, reverse, is useful. You want to understand the components, understand the loading of different components So we will, it's easy to go A times B, but it's very hard to do opposite. Okay. All right, let's take 5 minutes break. We have covered the most important things. If you see a slide with the right, this star, yellow star, it's the most important slide, okay? You only have maybe 5 or 6 across the whole semester. So once you see this start, you try very hard to understand, to remember. I hope after this will be easy, okay? Break. still miss one. Now, I want to consider if you have forgot about A and B, so you only get the product of AB, you get C. That's the matrix you observed, I gave to you. In our story, this is a shopping list. You and your roommate to buy things. But if I tell you, this is generated from the single cell data study. So this is a cell number 1. This cell number 2, okay? And this is a gene 1, gene 2, gene 3, gene 6. Our matrix will be larger, so you have thousands of cells from the single cell study. You have thousands of genes. The same story. What you observed is a mixed shopping list. You don't know what cell 1 is making internally. You don't know how many sandwich cell 1 is doing there. Another neighbor cell number 2 is doing. But you only have this comprehensive shopping list about a lot of components. These 3 genes, these 2 genes may be important to make bread, something else involved in the different sandwich type of making. So that's our problem. We have observed shopping list, the component numbers, but we need to figure out what they are doing functionally, functional group, and how strong each cell gets involved in which particular function in order to understand. So that's why the reverse problem to get from a C matrix to figure out A and B is important. Now we don't know how many sandwiches they're making, how many gene expression program each cell got involved. Maybe 20, maybe 50 major ones, and the more specialized cells, maybe only 1, because like insulin cell, this beta cells only make the insulin. So maybe it's only 1 component. It's very important for that cell type, but other cell type maybe have multiple function. Okay. So this is re-emphasize the problem and linked with what we are doing. Again, I hope you remember what I drew here. So if I have this, so this had to match, because here I have to be a square, basically. Okay, so if I have a 2 by 3, here have to be 3 by 6, and get 2 by 6. Okay, remember this framework. Now, if I reduce this to a vector, so it's only 1, then I should get a 1 by 6 results. Different color. So just as I said, so a vector and a matrix multiplication is a special case of matrix and the matrix multiplication, right? It's just one of the matrix become vector. So in this case, it's 1 by 3. So you get the product, another vector, 1 by 6, which here is 3 by 6 matrix. Okay. Now another special case will be, this is also a vector. This is a vector, another vector. So what's the product of this? The dimension of this product. will be 1 by 1. Okay? So this is vector to vector inner product. So as long as you have this arrangement, you can figure it out whether your end product is a single number or a rectangular matrix, what the dimension should be. Just remember to put it in this way. So I don't need to spend the time on this because it's very easy for yourself to figure out as long as you know how to do the matrix to matrix multiplication. Okay, now let's introduce this concept of rotation matrix and also this geometric implementation, interpretation of this. Now I use this example to show a matrix can be an operator, as I said. The squared matrix. A squared matrix in our case is 1, 2, 2, 4. So I can use this to multiplicate with a vector, which, for example, let's use this x as a vector as an example. So my x is 1, 1. So this is the vector. Okay, this vector is a data point in the space point to the position 1, 1, which is here. So this is my x. It's a vector, it's a data point. And the vector including the information about the angle, the direction, and also the length. First, can I do multiplication between A and X? So if you consider this as a two matrix multiplication, you have to make sure the inner dimension has to be matched. So what's the dimension for this? So this is a 2 by 2, and this one is 2 by 1, okay? 2 by 1. So it's possible to match its inner dimension because 2 by 2 times 2 by 1. Okay, 2 by 2 is here, and then you have 2 by 1. So this is 2 by 2, and 2 by 1. So this is A, this is X. So what's the product, the dimension of this A times X? So 2 by 2, 2 by 1. So the result should be in this position here. Should be 2 by 1, okay? So this should be 2 by 1, the position. So this is A times X. So now we consider A as an operator, is action on the X. The multiplication make X, because times A becomes the result A times X, which the dimension didn't change. So here is 2 by 1, your input. And output is also 2 by 1. Okay? So we did something on this data points. But this data point still, after operation, is still a vector, still a data point, still 2 dimension. So it's still in this space somewhere. Somewhere, A and could be here, okay? So if you know how to do multiplication to matrix, you can know, okay, this becomes, so 1, 1, and becomes 3, 6, if you do. So from 1, 1, 3, 6. Okay, so what does this matrix operator do to this input data point? The input is here, it was here. After you do this operation, it becomes longer and also changes the direction. From this position to there. So this operator rotates the direction of your data and also changes magnitude. In this case, make it longer and rotate to the left side. So a squared matrix can be an operator to operate onto the data points using multiplication and to rotate and stretch the length of the vector. So I hope you can derive this by yourself using matrix to matrix multiplication. It's just a special case. This is a squared matrix. This is a vector. Vector is data. Okay? So if you know how to do this, now we are introduce the concept which there's a special data point. Okay? So you can apply this to any points, any vector in this 2D space. This is because this is 2 by 2. If you have a 3 by 3 matrix, squared matrix, you can use that as a rotation matrix applied to 3D data points in 3D space. You can increase the dimension to n, still the same. Okay? You will rotate and stretch the vector. However, if I change this to 1, 2, and this is a special vector in respect to this rotation matrix operator. Why it's special? Because if you apply the same rotation, the operation matrix to this particular one, it only change the magnitude, not the direction. Because you multiply this to that, make the product 5, 10. 5, 10 is extension of this vector to the same direction. Make the vector longer, but not change the direction. So this vector in regard to this particular matrix is a special vector. This is called eigenvector. So this special vector is eigenvector of this squared matrix because it indirectly reflects what the feature of this operation is. This particular vector, the direction, tells you something about inside here, okay? How these four numbers in combined operate. When we write this into the equation, that will be, so A which is a squared matrix times this particular vector, which is specially selected. Eigen is a German, and it means the feature or standard feature, something like that. And this will be a single number times x. So this equation, if you plug in this A and this x, you will see, okay, that's true. Now we generalize this into a lambda. So this just indicates this is a single number, okay, scalar. So this formula gave you this particular equation to combine eigenvector, this particular number called eigenvalue, this is like a pair, okay? This is called eigenvector and eigenvalue for A. If you change a number here, your eigenvector and eigenvalue will change. So we start from an interpretation of geometry to introduce you this equation, which is based on the matrix and the vector multiplication, to give you some idea of what is eigenvalue and which is eigenvector. Okay, so this is another slide in here with a yellow star on it, okay? For now, you may have no idea why we need this, but in general idea is you want to use the value of this particular vector to learn the feature inside here, okay? You have no idea what the values in here, just like you have no idea about your components of those, although that one is not a squared matrix, but you can make that into a squared matrix. Once you have a squared matrix, you want to learn the relationship between those values, those elements inside here. You can borrow the eigenvector, so this information to tell something inside this squared matrix. Okay? For this lecture, we just leave this introduction of the concept. When we use it, we will use this equation later. We use this relationship later. Okay, so the rest of the lecture just quickly goes through some concept. There's no slide with a yellow star on it. So inverse matrix, so just in this formula, so if you have an inverse matrix, you multiply it by its original, then you will get identity matrix. So the trace, just add up all the elements in the diagonal position. So if you have a matrix, then this diagonal position will be position 1, 1, 2, 2, 3, 3. So you just add them up. So that's why you see this KK and the position where the index of X and Y are the same. So K, K, position, add them up. This is called the trace of a matrix. Determinant is a single value, okay? Basically it tells you the space that is occupied in the depending on how big the space dimensionality you are. Okay. Transfer. So so this operation, that A also considered as operation, so the volume new divided by the old. So after you transform this volume, and then what will change? This is a determinant. So this yellow star is here, okay? When we talk about eigenvalue and eigenvector system, we are dealing with squared matrix. So A has to be square. Has to be 2 by 2, 3 by 3, 4 by 4. If it's not a squared matrix, we cannot do this. Cannot find this, okay? You see here, only change length. What that means is this vector only change its length by a scalar value, so this eigenvalue here, after being operated or transformed by the M matrix. M is a squared matrix. Okay, so this is a couple of additional slides to re-emphasize the concept in a different ways, okay? Another numerical example. Here we are dealing with a squared matrix 2 by 2, okay? And there are two different eigenvalues with two different sets of eigenvectors. So if you plug lambda 1 with x1, it will satisfy this equation. Then you can find another, which lambda equals 2, and also with the eigenvector 2, 1. If you plug these two numbers in, you will get the equation satisfied. But you cannot max up with lambda 1 with x2. They're not a pair, okay? Not the corresponding pairs. So if you want to try, just calculate this manually. When you have has to be a squared matrix. And also, for this example, it's also a symmetric matrix. Symmetric means if you do transpose of x, you will get exactly the same thing. It's squared, so but you transpose it, you have the same thing because they are symmetric. There are good features for this symmetric ones because if you have a symmetric squared matrix, you will for sure have this real values of these eigenvalues, real numbers. Otherwise, it could be a complex number, which is difficult to handle numerically. So we really like symmetric squared matrix. So we calculate eigenvalue, eigenmatrix vector using computer. There are data science method which dedicate to see, okay, given a squared matrix, how can we calculate numerically very quickly on computer to do those. But if you want to try a small example, like a 2x2 matrix, you can manually calculate its eigenvector. I know it will be very hard for 3x3, and nobody try to find a 4x4 matrix eigenvectors. So this is called, one of the application of this is to, this relationship is to do the eigen decomposition. Remember I showed you the sandwich examples. If you have A matrix multiplied by B, you get a C, but you can hardly decompose the C back to A and B. Here gave you possibilities. If the C is a squared matrix, for example, if you have a C like A, so this A can be decomposed using this equation into, so you put this lambda in here. You basically move X from this end to the other side of the equation, but take reverse. So you need to put this back here. So this formula is the same as this. They are equal. But this gives you some idea. You decompose A into this, into 3 parts. Okay, that is not exactly as we decompose the sandwiches, shopping list into sandwich and what you, how many you want to make. But it gives you some way to decompose the squared matrix. You make this into 3 elements multiplication. One is a vector and its inverse, and another is a single number. So in our case, the number 5, so it's very easy to make that decomposition. Then you found the 2 vectors. You can some kind of decomposition, okay, operation. So that's what shows here, okay. I think that's all enough, and also very important for you to understand matrix multiplication, how to do it, and the matrix multiplied to a vector is a special case for the MM matrix to matrix multiplication, and also squared matrix as an operator. When you multiply it to a vector, you change its direction and change the magnitude of a vector. So that's also, if you have a special case, only change the length, but not change the direction of your vector you operate on. So that vector can be considered as an eigenvector to showcase the feature in the squared matrix. Also, if you apply eigen decomposition, you can decompose square the matrix into different parts utilizing this eigenvector definition. Okay, very quickly go through the cell ranger. Do you have a quick question before we move on? It's fine? Good? Okay. You will have a homework to describe how to do the multiplication between two matrices. Just find a way to show me, okay, you know how to do it. If I gave you A and B, two matrices, okay? There are many different ways you can show me you can do it. The way I will let you to take a video, okay, to explain to me. Yeah, it doesn't matter. Whenever you want to submit. No, not very important. But as long as you know, okay, sooner or later. So this is a controller, and you load your samples in and get captured by all your droplet, and also inside the droplet there are gel bead. So I haven't done this, but they're all preparation of the samples. You need to make sure all the channels, because you have 8 different channels here. You don't want to load some empty bubbles in there, but make sure they all thoroughly penetrated and loaded some amount, even though they are isolated. You want to make sure you have live cells instead of dyed. You pick some samples from your cell suspension and check after, put some dyes in there. And this is a barcode system, and you have a 10x genomic barcode which cover the gel bead is unique for each bead, and then you have UMI. Each UMI is different. There are many, many different UMIs. Each gave you a label for that particular mRNA. This is a setup of the gel bead, and with the UMI indicated in this orange barcode, and everything will be sequenced together. So you will get a sequence with one end with a 10x genomic barcode, UMI, okay? So you will use that information to tell this particular transcript is from the cell which labeled by the 10x barcode, and from that particular gene, which copy, UMI, just one copy. So this is a library preparation for sequencer. You have forward strain, reversed, the read 1, read 2, with this information in there. So that will be your forward, and this will be reverse. Some sequencing details. Inside here, there are sample index, 6 base pair to tell you different batch of samples. Then 10x genomic barcode is a 16 base pair to cover enough cells. The UMI is a 12 base pair sequence. Read 1, read 2. This may be updated, out of date, because they have a new version. You may not require read 3, so that's why we tried some examples with R1, R2 reads, which R1, R2, and also have an index. But nowadays, they probably don't need that. The R1, R2 is enough. So this is a barcode system. There 2016 original version, 16 base pair barcode. This is covered by on each of a bead. So you can, if you open this file, you can see this barcode.
from this AACC, the first one, until 737,000 until end. Okay, so this gave you cell separation. And for each of this barcode, you will be given this 9 base pair and 12 already in version 3, 12 base pair of UMI. So each of this UMI will be individually labeled onto each of this barcode. This is a whole system. So in terms of software, and then inside the controller, there's a system to run this and create GM, which is the droplet. And the cell ranger is what we use in the cloud computer to get this sequence mapped into reference and label this transcript is from this gene. So it gives you the name of the gene for each transcript. And also separate cells by the 10x genomics barcode in so group of cells to get the matrix. And then they also have this Looper browser and supposed to give you the visualization of the cells, but we use our own MATLAB version. So the pipeline, something like this, and this is a R1, R2 code sequence, the FASTQ from the sequencer. And this is command line. If you don't use 10x genomics cloud service, you have to install Cell Ranger on your Linux machine and to run the count and to provide reference. This is reference, the human reference, and to provide where you put your FASTQ files. Okay, let it run. You will get exactly the same results as you run on the cloud. So we don't have to bother to set up our own Unix machine, Linux machine. And there's other comments, for example, aggregation of different samples. This is to explain how the UMI work. So these color ones are indicated by the cell barcode, 10x barcode. They are different so that you can separate cell 1, cell 2, cell 3, and there are transcripts, mRNAs from these different cells. And then you use UMI to indicate, okay, this is the cDNA, this is mRNA from this gene, that's this particular copy has been labeled. And this is a separate copy labeled. That's 3 different UMI label, 3 different genes. Well, in this case, in cell 3, the 2 UMIs labeled to the same ACTB gene, basically 2 UMI in the same mRNA. So in this case, we will count the number of expression of the CTTB, ACTB, only 1, because this UMI duplicates by the PCR amplification on the same mRNA, same molecule. So we only count 1 for this gene. Well, it's different from this. This number 4 cell, same gene, but they have different UMI, label different molecule, this distinct transcripts. So all those are from the same gene. That's a biological copy of that. It's not created by the amplification of PCR duplication. So if you still have no idea what this is about, come to, well, you have to figure it out. Okay, who haven't got the idea, UMI, how UMI work? Still have some trouble? Yeah, I have an office hour. If you make appointment to come here, my student can explain to you. We don't have time. I probably spend some more time on Thursday in front of the computer, but this is a report the cell ranger will create. It will tell you in your sample how many cells have been sequenced or get information from, and how many genes per cell. Okay, you need to have some feeling about those numbers. For nowadays, we have 10,000 cells in general, but this is a little bit higher for me number. And also there's saturation curve. You will see how many barcodes, and each barcode indicate a non-empty droplet. And inside there, you should have enough UMI count. So this cell have this amount of the UMI count. If you have a very low UMI count, basically that tell you this droplet is empty, or the cell maybe not there, and some contaminations in there. You get some UMI counts from neighbor cells maybe. So you need to have enough barcode. So the number of cells is 10,000. We need 10,000 here. Have enough UMI counts for those cells. And you expect to see a quick drop. That's a separation between droplet with cell and without. You should have a very clear separation in this end. If you have a very smooth go down, that indicate their spectrum. Some cell have half, some droplet have half number of the UMIs. So maybe indicate that your sample is some kind of mix of the contamination, or all the neighbor cells have a shared mRNAs in the environment, in the neighborhood. So not a good indication. You need a very clear knee shape of this drop down of this curve. Okay. That's all for today, and very important, you have a foundation for linear algebra and know how to do matrix multiplication, and have some idea what an eigenvalue, eigenvector is. Okay? Okay, good. See you Thursday.
Thank you.